Hey everybody, John Marshall from the 2018 Ruesch Symposium, Fighting a Smarter War Against Cancer. I'm joined with incredibly smart people. That's the secret to a good symposium, is invite people that know more than you do, which is a lot of people. But I want to kind of go through each of these folks. Our topic is tumor targets. I have trouble as a busy clinician keeping up with what's out there and what's going on, and when are some targets going to be druggable, what are the new targets that are out there. And so these guys have done a brilliant overview of some of the highest level, furthest along, I would say, novel targets. And Eric Collisson flew all the way across the country from California to join us today. So maybe start with you a little bit. I love this topic of homologous repair, BRCA, some people think. So give us a quick summary of what you discussed and sort of its impact on day-to-day care for patients.
Sure, happy to. So one of the biggest emergences in oncology these days has been a new class of drugs called PARP inhibitors. We're learning how to use those, and in the gastrointestinal oncology world, as you know, we're kind of following on the shoulders of giants, and our breast and ovarian colleagues have really developed these drugs.
Better funded giants.
Better funded for now.
Let's be very clear.
So stay active. We can, as we know, our GI cancers kill more Americans than breast and ovarian cancer combined, but there's no competition.
Yeah.
I spoke today about how we're trying to use these drugs in pancreatic and other gastrointestinal malignancies, and what kind of footholds we have to teach us in whom to use them and when not to. And one of the things that we had in ovarian cancer was just you're born with a mutation. It's increasingly in a family that we identify through 23andMe and very consumer-driven things, but definitely in the medical clinic as well. And that made it pretty easy to find those patients to develop that drug. In the GI cancers, they're rarer mutations, but they're possibly in the low double digits, which in pancreas cancer, it could make a huge impact. So we're increasingly looking to use these drugs in pancreas cancer, other GI malignancies that have evidence in their genome of a disordered repair machinery. So like we talked about in the talk, these are genomes that when we sequence them in the lab, they're clearly abnormal, but getting some structure as to the way they're abnormal has focused our attention on a subset of patients with this BRCA-like signature.
So some of the basic science stuff that's come out about this, there's a list of mutations or abnormalities that get you in the BRCA in this category, but then when one of our fellows actually did a big nationwide analysis using Caris database, many of the lists that were on the basic science list, if you were not found at all in the human list.
Right.
And so how sure are we that we can kind of define this BRCA-ness using molecular profiling?
And yeah, this has been a real last mile problem, is how they put it in technology. As we know, there are a few big players that when they're mutated, they clearly cause the cancer and they cause the phenotype.
And the therapies seem to work in that subtype, so it's a nice match all across.
So we've gone kind of soup to nuts on that cycle. The problem has been that there's not a direct correlation as to things that score in fruit fly assays and C. elegans assays that are clearly put this protein on the pathway. That doesn't mean that it's necessarily a player in cancer in humans. And so there's been about 30, 40 years of data looking at these genes in Fanconi's anemia and other human diseases. That doesn't mean that when mutated, they get a cancer out the back end. Sometimes the cell just dies, for example, and it doesn't go anywhere. Or sometimes there's a lot of redundancy in the human that there isn't in the fruit fly, for example.
One of the great things you brought forward at the end of your talk was about clinical trials that are looking at this in GI cancer space. We're doing more and more molecular profiling. NCCN guidelines say we should be. We're finding these. Getting access to these medicines is an issue, particularly now approved PARP drugs.
Right.
Can you give me a little sense of, if I'm a doc and I find one of these positive results in a pancreas cancer, say, what do I do with that?
Yeah, it's a great question. You're probably going to be spending some time on the phone.
With Aetna, I'm taking it.
Yes, exactly. Well, we do. That's why it's important to accrue and to participate in clinical trials. The POLO trial by AstraZeneca is moving forward and almost closed, looking again at that germline pancreas cancer variant. However, we both expressed our interest in finding somatic variants, mutations on these panels, for example, that we might not have expected. Now you can make a fair but non-guaranteed case that there may be benefit to these patients. Certainly in pancreas cancer, our group, and I think Michael will agree, we rely on oxaliplatin, or the platins we have in our clinical regimens already, to do a lot of that DNA damage heavy lifting. How that'll compare to PARP inhibitors remains to be seen.
So if I'm choosing between gemabraxane and a FOLFIRINOX, if I know this up front, I'm going to use an oxaliplatin base.
That's the thought. We actually think that these cancers may be more sensitive to Abraxane as well.
Yeah.
So it may go either way, but we look at a good response as who cares why you're right.
Perfect. Dr. Channing, you just gave an amazing talk on RAS, and you know, I think about the number of patients I see with a RAS mutation. I just go in, and we know them all now because we're profiling all of these patients. And we're finally, it sounds like, making a little headway. Give us a quick summary of your discussion today.
Yes, so for better or worse, we have struggled with this issue of trying to target RAS for more than 3 decades now. And I think I've suffered through every second of that sad history. But I believe that we are now making some significant inroads. And I would cautiously believe that maybe within the next 5 years, we're going to have some therapies that will be effective against RAS mutant cancers. And in my presentation today, I talked about one discovery that we made, which was somewhat fortuitous in that we weren't expecting this. We discovered that the combination inhibition of a key RAS pathway, the MAP kinase pathway, elevated a metabolic activity which would seem to drive cancer, and that did not make sense to us. But rather than not getting confused and saying, what do we do with this confusion, we said, let's understand it, and let's potentially exploit it for therapy. So at the end of the day, we found out that treatment of pancreatic cancer with an inhibitor of the MAP kinase pathway resulted in cancers that became much more dependent on a metabolic process known as autophagy or autophagy. And this then made that cancer much more vulnerable to inhibitors of autophagy. And so now we have transitioning these findings to the clinic of combining an inhibitor of the MAP kinase pathway together with an inhibitor of autophagy, hydroxychloroquine.
Provoking a bit, if we could really control the RAS pathway, I mean, if we really could control it pharmacologically, how impactful do you think that will be in the clinic? How strong of a driver is that in our patients with GI cancers?
So if we believe that mice somewhat model humans, the data from studies in mice, either genetic studies or pharmacologic studies, would say that if we can impact and disrupt RAS function, the consequences on GI cancer growth, colorectal as well as pancreatic, will be very, very significant.
So this would be bigger than immunotherapy, in my opinion. I mean, because just the frequency of these mutations, the commonness of these diseases.
Yes.
So, but there's a lot of hope out there. And so this is one of the things I'm trying to govern on the front lines with patients, is they're watching this space. I mean, they're smart enough to be knowing this.
Yes.
And they're starting to see word. I think there was a case out at City of Hope who's on one of these phase 1 studies who has an early pancreas cancer that on 1 month of therapy has already had a plummeting marker and a responding tumor. You know, is this real? You think this is real?
Yes, so I think that the successes we're starting to see are an indication of real success in the long run. One caution I would say is that it's not going to be a single anti-RAS therapy that will be effective for all RAS mutant cancers. Because of the genetic complexity of cancers from patient to patient, we're probably going to have to develop multiple RAS therapies, and that each approach will be matched with different subsets of patients, which then, of course, requires that we have an ability to identify beyond a RAS mutation what makes this population responsive to this anti-RAS therapy versus another anti-RAS therapy. But I believe they're going to be coming down the line within the coming years. And so after many years of failure, I'm very cautiously optimistic that we're finally going to accomplish what we thought was impossible.
Last night at our Luminaries event, Joe Biden chided us a bit and said, we have to collaborate. The only way we're going to move the bar is if we collaborate. And I was so pleased to hear that you had some parallel science that you guys are now working together on. And it seems to me that this community targeting RAS is as coordinated in some ways, yes, competitive, because we all want to be the one on the first author on the paper, but at the same time knowing that this is affecting so many people.
Exactly.
How hard is that to make those collaborations? How hard was that to say, okay, we'll play together?
So it actually is it's pretty easy, I must say. I think in particular the pancreatic cancer field, and Eric might also weigh in on this, it is a field that has put the patient first and said, this is the important thing. And in order to accomplish this for the cancer patient, the best thing for us to do is work together, because science moves very quickly and we cannot be experts at everything. And so we would need to work together to bring our respective talents together to try to achieve something that individually would be much more daunting for us to try to achieve. So I think the teamwork aspect of cancer research, in particular in the pancreatic cancer field, is wonderful to see.
One of our great partners at the Ruesch Center, as also one of yours, is PanCan. I mean, they've done such a good job of transforming the advocacy around that, both patient-facing as well as science-facing, and has really become one of the leaders in funding. And I think you both have support from them. Been critical. Is that part of their deal? You have to collaborate if you're going to use their money?
They encourage it, and I want to really shout out both to you and to PanCan. One of the distinctions is PanCan has actually reached out and pulled in basic scientists like Channing. I mean, Channing has been doing the good work here for 30 years, and it wasn't until a few years ago that he was really brought in, and I think his science has exponentially improved. Well, it was great before, but it's so much more translatable now. We got him working on KRAS instead of HRAS. We got him working on people in mice instead of just cells, and it's a great example of an audience that has really demanded the basic science.
It's back to my comment earlier about giants. I mean, they're well funded. You follow the money. So if you get some good scientists are eager to solve these problems, but they have to have money to work with it, and so we're all trying to work together to move that forward. Michael, I've not left you last for any reason other than you've got the coolest stuff to talk about. Maybe, I don't know. This is big common things that would be dramatic breakthroughs. Your focus today, at least for the purposes of our meeting, is on rarer abnormalities and how important it is to find those and the drivers that are out there. So maybe talk a little bit about what you're going to focus on today.
Yes, so I think in general, we are really fortunate that we are witnessing now the translation of the idea of personalized medicine into clinical reality. And we are able now to address challenges that we have been looking at for many years. Among those are the very common molecular changes that we see in cancer, like RAS mutations. But when we use next generation sequencing approaches, what we see is a very complex genomic picture in almost all gastrointestinal cancers, actually, all solid tumors, where on one hand, there are these recurrent mutations, P53, RAS, and so on, that we find in very many cases. But then there is this long tail of aberrations. That's why I call the talk Chasing the Cancer's Tail, of very low frequency alterations that when you look closer can have a major clinical impact. And so that is a different type of challenge. It is how do we make sense out of these highly variable mutations in a way that we deliver solutions for individual patients in a situation where the total number of cases we have is very small. But I think we're making really progress in this regard, and I think that is most prominently highlighted by a development that just happened this week, that we see now for the second time a drug being approved for cancer treatments based on the presence of a molecular lesion and not based on a cancer type. And this is the approval of larotrectinib, which is an inhibitor of the NTRK kinase, which it turns out in a small number of cases is activated in cancers through the generation of what we call genomic fusions, where parts of one gene are being used to drive a highly cancerogenic, oncogenic protein expression, in this case NTRK, that leads to cancer development and maintenance. The advantage of these kinases is that we can target them with very specific, highly potent inhibitors. But the challenge is, for example, NTRK mutations occur in 1% of the cases. It turns out that when you do this efficiently, you induce responses in 80% of the cases.
Yeah.
Numbers that we have never witnessed before.
Tumors melting too. I mean, the pictures
It's
are dramatic. Yeah.
Rapid and then with long duration of response.
So we're adding to the pile, right? We had the same issue with MSI, right? So we had a cross-disease approval. Some disease groups, to be nameless, said, oh, we've got a new drug for our new disease, when in fact it's maybe 1% on a good day. So we have to temper our enthusiasm and the press's enthusiasm for these discoveries against the reality that you're unlikely as a patient to be one of these cases. But then you start piling them together, right?
That is exactly what's happening. That we address one very small slice of the whole challenge, but now we're seeing them being added together, and all of a sudden you get a significant slice of the pie. Yeah. And so in gastrointestinal cancers, we see certain themes developing, targeting mutations or other activating alterations that affect signaling pathways. And the other theme we just heard about is BRCA-ness, where many of these mutations occur at very low incidence. So that slice is definitely growing. And the big question is, and the big challenge is, how do we demonstrate that targeting one of these particular low incidence mutations that could have high clinical impact, that this is really happening? Right. And one approach is through basket trials. And among those is the most famous one is NCI Match. Actually, Caris has been involved as one of the testing laboratories for this trial. And just recently we learned that one of the arms was positive. Inhibition
Finally.
Exactly. We were kind of, it was a long
frustrated
period of waiting. But it turns out targeting AKT1, again, across multiple cancer types, seems to be efficacious.
Yeah. So oncologists out there, should they be ordering these tests? And what I hear a lot of is they get very frustrated. They get back P53 and RAS, and right now you can't do anything with those. And a lot of other things they've never heard of, and some unknown significance BRCA mutations, which makes the patient think they have inherited cancer syndromes. So where are we with this, and where are we? I would actually also have you comment a little bit on the changing environment around reimbursement for this, because our world is dictated by acceptance by the payer.
So when you look at these enormous response rates to NTRK inhibitors, but then also look at cases, as I will show in my talk today, where we targeted a patient with colon cancer and a POLE mutation, which is, again, a 1% kind of event, but is associated with a great sensitivity to immunotherapy. Yep. When you see those cases, you realize we are now at a point that we need to move molecular profiling right to the very beginning, when we start charting out the path forward for our individual patient. because we will be able to either now increasingly choose FDA-approved treatments, but also line patients up for clinical trials where they would fit in best. And it's important, actually, especially when it comes to these fusion events, to also then, when you order this kind of modern profiling, to order profiling approaches that they have the highest chance to pick up these events.
So this is back to the knowledge base, is that when I order one of these things, I think I'm getting it all, right? But I don't really know what I'm missing.
Yeah.
I mean, I know, but I often don't know what's not in there.
Yeah, and it's especially true, I think, as a practicing oncologist, when there's a hand carry test, right? You know what you order and you know what's on the test you use. But patient Joan Smith comes in from another hospital because you're the expert, and you've never seen this test before.
Happened to me the other day.
And you often have a photocopy that's kind of scanned sideways, and you can't even read the font.
Right.
Let alone what genes are tested, what the negative predictive value is, positive predictive value. So these are real issues that I think none of us really learned in medical school as oncologists. We're not lab directors, and we're not. We don't really understand the nuances that go into this.
Well, and the quality of the test, too. I mean, what you're saying
you
is...
don't know what you don't know.
You know if it's a crummy CAT scan. I always like this, you can look at a scan and go, we need to repeat that.
Repeat the scan.
But when you get a molecular test back, you don't always know if it's a good test or not. Yeah.
Correct, and I think the answer is having thoughtful people in the industry who are, not just technical. They're clinically prioritized, which I'm encouraged a lot are.
But choosing the right technology to look for it is, I think, critical. For example, fusion events are best detected at the RNA level because this is the final product of this essentially artificial gene. And it might be configured in different ways, but at the end it will result in an mRNA product that will lead to the activated protein. So looking at the RNA level is critical for this. And so there are a number of these examples where using really the best technology to detect these things is really critical. The other aspect, and we built this at UCSF initially, but we'll try this to do this with a bigger scheme now with CARES and trying to roll this out nationally, is providing doctors access to molecular tumor boards. Because I have to say, we have learned tremendously through this mechanism at UCSF, and by helping oncologists to interpret these complex findings, I think we can deliver better care to our patients.
Yeah, I think on some level, back to the radiology equivalent, if it's like sending a doc the images and saying, here's the CAT scan you ordered, and not the report. And where what we need is that interpretation with the raw data. We often look at our images after we look at the report because we can interpret those, but that's part of the learning curve. But thank you guys very much for making the journey here from far away, and we appreciate your sharing your thoughts with us today.
Thanks.
Thanks for having us.